Assalamualaikum and hi. My name is Aisha Suhana binti Muhammad Suhaili and I will presenting a journal entitled Genomic and Ecological Attributes of Marine Bacteriophages Encoding Bacterial Virulence Genes. Firstly, introduction. Do you know about 10 to the power of 31 particles bacteriophages are the most abundant biological entities on the earth? Bacteriophage genomes undergo frequent lateral gene transfer. These phage encoded genes that can be shared with microbial hosts and fixated under selective pressure. Viral genome size constrained by the capsid volume and mutation rate. As a result, it condenses genomes with frequent overlapping open reading frame. Most of these genes bring adaptive advantage to the viruses. The expression of the phage gene during infection confers new functions and also modulates existing host functions. Besides, bacterial virulent genes are often carried by temperate phages and lysogenic conversions. For example, prophages inserted in the genome of the coral pathogen Vibrio corallitis show increased nucleotide sequence identity and similar gene organization with virulence gene encoding Vibrio choleraphage, thus suggesting that lysogenic conversion caused coral disease. This study suggested that phage-mediated bacterial virulence contribute to pathogenicity in many marine diseases, but the phage-encoded virulent gene is still unknown. Next, methodology. Sesi viral genome dataset. Viral metagenome from the coral reef is analyzed and samples were collected from the reef boundary layer in Southwest Atlantic, Central Pacific, Hawaiian archipelago. DNA was tested from the bacterial 16S by PCR and the negative sample were analyzed. Viral and abundance from each sample were determined by epifluorescence microscopy and flow cytometry. Second, viral genome prediction. Database of viral genomic sequence was compiled, including viral genomes from reef sequences. Second, uncultured marine viral genomes assembled from the multiple studies. The selection of viral genome sequences and exclusion of potential bacterial contamination as follows. Firstly, contigs derived from the assemblies were combined and less than 2,500 best pair is removed to decrease the false positive chance. Second, coding DNA sequence were identified with prodigal within POCA. Protein sequence were queried against the NCBI-NR database for annotation using diamond. Third, source of phage genomes are first NCBI sequence database, second Cosmic Libraries, and Zero Sorter. Fourth, dice coefficient score was used to estimate the distance between contigs and organized into a phylogenomic framework. Host prediction based on homology matches against bacterial and archaeal genome, homology matches of the viral genome sequence against Saharan Ocean microbial contigs, and transfer RNAs identified in viral genome sequence. Third, virulence genes assignment. Viral protein predicted from the contig open reading frame were queried against the curated database using +P. And lastly, statistical analysis was done by non-metric multidimensional scaling to test the relationship between bacterial density and host taxonomy. Results: 28,483 viral genome sequence presenting viral assemble of the viral genome sequence composed the viral community in the coral reef. 1,149 viral genome for 2 to 4% of the viral community encoded at least one bacterial virulent gene. About 70% of the open reading frames in this genome encoded putative protein with unknown function. Figure 3 shows a common characteristic of phage genome. Photobacteria genus Vibrio are common marine pathogen. Lysogenic conversion in coral reef Vibrio and extends that to the other bacterial group. Thus, lysogenic conversion mechanism widespread among marine pathogens. This study provides a platform in the search for potential phage-mediated mechanisms causing marine disease of unknown pathogens such as black band disease, white plague, white fox, and stony coral tissue loss. Conclusion: this study shows that coral reef microbialization is associated with the rise in phage-encoded virulence genes that enable bacterial recognition and invasion of metazoan, thus lysogenic conversion as a host polymicrobial disease contributing to coral reef decline. That's all from me. Thank you.